Now let's go on to look at the T cell response. At the same time as the B cell line has been actively responding to antigens, the T cell line has been similarly engaged. B cells are all about getting antibodies into the fluid compartments to fight invaders on a humoral level. However, the response of T cells are all cell-mediated immunities, so we think of it as the cell-mediated response, or CMI. It requires direct involvement of T lymphocytes throughout the course of the reaction, and the T cells require some type of MHC recognition before they can be activated. All of them have cytokine production with a broad spectrum of effects, as we'll see shortly. And rather than making antibodies to control antigens, the T cells stimulate other T cells, or B cells, or phagocytes in their immune responses. So we've explored activation of T cells, but now let's look further into how they differentiate into the various subsets of T cells, because there are a lot of different types of T cells. T cells, as you know, mature in the thymus and are primed to react with antigens as they sit in their lymphoid organs. They recognize antigens only when they're presented in association with an MHC carrier molecule. The T helper cells come in basically two forms. They have cells with the CD4 marker on them or cells with the CD8 marker on them. Those with CD4 receptors recognize endocytose peptides that are on MHC2 receptors presented by the antigen-presenting cells. Those with CD8 receptors recognize peptides on MHC1 receptors. These are found, if you recall, on all nucleated cells, so they often represent self. The T cell is sensitized when an antigen and MHC complex is bound to its receptors. The activated T cells then transform in preparation for mitotic divisions and differentiate into one of the different subsets. Here you can see the different subsets of T cells. You'll recognize that all of the T helper cells are CD4 type cells, and CD8 is a cytotoxic T cell. Well, they're very different in their functions. Essentially, all of these CD4 cells are helper cells, even including the regulatory cells, but we'll cover that here shortly. The CD8 cells are cytotoxic T cells. They're highly destructive cells. When we look first at T cells, we recognize we're making a decision of whether they're CD4 or CD8 cells. There are two distinct lineages that will arise from these different T cell types. In here, we're looking at the T helper cells. So these are the ones that arise from T cells with the CD4 marker on them. They play a central role in regulating immune reactions to antigens, but they're also involved in activating macrophages. They can either directly contact macrophages, or they can contact them indirectly by releasing cytokines like interferon gamma to activate the macrophages. Either way, the antigen-presenting cell is going to release interleukin-2, and that interleukin-2 will stimulate development of a number of different types of T cells, each of which you can see here. Some T helpers will secrete other interleukins that stimulate various activities of B cells. So when they're stimulated by an antigen MHC complex, as you see here, they'll differentiate into predominantly T helper 1 or T helper 2 cells. The T helper 1 cells are responsible for secreting substances that influence the B cell differentiation and enhances the antibody response. If they become T helper 1 cells, then we're looking at macrophage activation. T helper cells are the most prevalent T cells in the blood and lymphoid tissues. They constitute about 60% of cells in those regions. And it's depression of these T cells, the ones with the CD4 receptors, caused by HIV, which accounts for the immunopathology of AIDS. Shutting down these T helper cells pretty much shuts down the immune response, hence the name acquired immunodeficiency syndrome. Now, the regulatory T cells, or T sub R cells, can be broadly considered to be in the T helper class. Because they arise from a CD4 cell, they're included really in this main group of T cells. However, most people consider them to be in a class of their own. They do act to control inflammatory processes and prevent autoimmunity. They also prevent inappropriate targeting of a resident biota. The third type of T cell were the cytotoxic T cells. Cytotoxicity is the capacity of certain T cells to kill a specific target cell. These are sort of the worker bees of the T cell population. This property of cytotoxicity is a really powerful tool that accounts for much of our immunity to foreign cells and cancer cells. Keep in mind, we're down here now looking at the T cells with CD8 receptors on them. To be activated, these T cells must recognize a foreign particle complexed with a self or MH1 receptor protein. When they are complexed with this MH1 receptor protein, they have a direct attack on that target cell. They become activated and they go right to work. They're able to actively destroy these target cells through the secretion of perforins and granzymes. Perforins are proteins that punch holes in the membrane of the target cell. This causes the ions to leak out. And granzymes are enzymes that attack proteins inside the target cells. So after the perforins have punched holes in the cell membrane, then the granzymes can enter and destroy the proteins of the cell. This causes total destruction of the cell, or apoptosis. So we have three different T cell types: the T helper, the T regulatory, and then cytotoxic T cells. This figure summarizes all the ways in which they work. Now, something to consider before we move on is how the B cells are involved in the T cell response, because remember, there is some overlap between these two divisions. First of all, we know that the T cells become activated to become plasma cells by cytokines that are secreted from activated T cells. We also know that those activated regulatory B cells can regulate the degree of response from T cells. This is this feedback reaction here. So although the humoral response and the cell-mediated response are very different, they definitely act on each other. Before we move on, let's consider one last type of cells, the natural killer cells. They're related to T cells, but they lack the specificity for the antigens, so they're not technically part of the specific cell-mediated immunity. However, they do destroy cells by similar mechanisms, and they are acutely sensitive to the cytokines released by these cells. Natural killer cells circulate through the spleen, through the blood, and the lungs, and they're probably the first killer cells to attack cancer cells and virus-infected cells. However, again, they lack the specificity of antigens. They don't need the MHC proteins or antigen presentation in order to be activated. Before we move on, let's watch a couple of animations. First, how cytotoxic T cells work. And then let's have a summary of the whole immune response.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class I MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class I MHC. The cytotoxic T cell then releases cytotoxins which induce apoptosis in the infected cell and perforin which causes perforations in the cell membrane. Self-proteins are also degraded and the fragments presented on the surface via class I MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
And now a summary of the entire immune response. Hopefully this one helps it pull it together for you.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
And then before we leave this section, how about a quick concept check? What type of cells are most important for helping B cells proliferate and differentiate? CD8 variety T cells, CD4 variety T cells, cytotoxic T cells, or killer T cells? Well, really, we're reviewing the functions of these different T cells. So this is a great question. CD8 T cells become cytotoxic T cells, so they're not involved in B cell proliferation. We can cross those off the list. Cytotoxic T cells, because they're a result of those CD8 T cells, they're also not involved in that process. So now we're left with killer T cells, which had nothing to do with the B cell response. So it's these CD4 activated T cells that are involved in proliferation of B cells.